Probability distributions, binomial, Poisson, and normal. Probability distributions describe how the values of a random variable are distributed. They are fundamental in statistical analysis and are used to model various real-world processes in biological, ecological, and zoological research. Among the most important probability distributions are the binomial distribution, Poisson distribution, and normal distribution, each of which has distinct characteristics and applications. Binomial distribution. The binomial distribution models the number of successes in a fixed number of independent Bernoulli trials, where each trial has only two possible outcomes, success or failure. It is used when there is a fixed probability of success in each trial and the trials are independent. Formula for binomial probability. The probability of obtaining exactly k successes in n trials is given by Px equals k equals nk pk 1 minus p n minus k. Px equals k equals k n pk 1 minus p n minus k. Where Px equals k. Px equals k is the probability of exactly k successes. N is the total number of trials. K is the number of successful outcomes. P is the probability of success in a single trial. Kn is the binomial coefficient, calculated as n. N minus k. N minus k, n. Key properties of binomial distribution: the number of trials n. N is fixed. Each trial is independent. The probability of success p. P remains constant for all trials. The binomial distribution is discrete, meaning that k can take only whole number values. Applications in biology and zoology. Genetics, probability of inheriting a dominant or recessive allele in offspring. Epidemiology, probability of a certain number of individuals getting infected in a population exposed to a disease. Ecology, modeling the probability of a species being present or absent in a given area. Example calculation. A geneticist is studying a population where the probability of a certain mutation occurring in an individual is 0.2. If 5 individuals are sampled, what is the probability that exactly 2 of them have the mutation? Px equals 2 equals 5 2 0.2 2 0.83. Px equals 2 equals 2. 5 0.2 2 0.83 equals 5 2 5 minus 2 0.04 0.512 equals 2 5 minus 2 5 0.04 0.512 equals 10 times 0.02048 equals 0.2048 equals 10 times 0.02048 equals 0.2048. Thus, the probability is 0.2048 or 20.48%. Poisson distribution. The Poisson distribution models the probability of a given number of events occurring in a fixed interval of time or space, assuming that the events occur randomly and independently. It is used when the number of occurrences is rare but can happen many times over a large period or space. Formula for Poisson probability P. X equals k equals e minus lambda lambda kk. Px equals k equals k. E minus lambda lambda k. Where Px equals k. Px equals k is the probability of observing exactly k events. Lambda is the mean number of occurrences in a given interval. E is Euler's number, approximately 2.718. K is the actual number of occurrences. Key properties of Poisson distribution: events occur independently. The probability of an event occurring in a small interval is proportional to the interval length. The expected value, mean, and variance are both equal to lambda. Applications in biology and zoology. Ecology, modeling the number of rare species found in a specific area. Epidemiology, number of new disease cases in a hospital per week. Genetics, number of mutations occurring in a DNA sequence over time. Example calculation. If an area of 1 square kilometer has an average of 4 rare birds appearing per week, what is the probability that exactly 3 birds will be observed next week? Px equals 3 equals e minus 4 4 3 3. Px equals 3 equals 3. E minus 4 4 3 equals 0.0183. 646 equals 6. 0.0183 64 equals 1.17126 equals 0.1952 equals 6. 1.1712 equals 0.1952. Thus, the probability of observing exactly 3 birds is 0.1952 or 19.52%. Normal distribution, Gaussian distribution. The normal distribution is a continuous probability distribution that describes symmetrically distributed data. It is the most commonly used distribution in statistical analysis and has a bell-shaped curve. Probability density function f. X equals 1 sigma 2 pi e minus x minus mu 2 2 sigma 2. Fx equals sigma 2 pi 1 e minus 2 sigma 2 x minus mu 2. Where x is the variable, mu is the mean of the distribution, sigma is the standard deviation, pi is approximately 3.1416. E is Euler's number, 2.718. Key properties of normal distribution: the curve is symmetric around the mean mu. The mean, median, and mode are all equal. The total area under the curve is 1, representing 100% probability. Approximately 68% of data falls within 1 standard deviation, mu plus or minus sigma. Mu plus or minus sigma, 95% within 2 standard deviations, and 99.7% within 3 standard deviations. Applications in biology and zoology. Human body measurements, height, weight, and blood pressure follow a normal distribution. Physiology, IQ scores, reaction times, and enzyme activity often follow a normal pattern. Ecology, distribution of species traits such as body length or wingspan. Example calculation. If the body length of a certain fish species follows a normal distribution with a mean of 30 centimeters and a standard deviation of 5 centimeters, what percentage of fish are expected to be between 25 centimeters and 35 centimeters? Using the 68 to 95 minus 99.7 rule, we know that 68% of values fall within one standard deviation between 25 centimeters and 35 centimeters. Therefore, 68% of fish will have body lengths in this range. Solved previous year question. Question. Which of the following distributions is best suited to model the number of bacterial colonies on a petri dish, assuming the colonies appear randomly? A. Binomial. B. Poisson. C. Normal. D. Uniform. Answer. The correct answer is B. Poisson, since the appearance of bacterial colonies is a random event in a fixed area, which follows a Poisson distribution. Multiple choice questions MCQs. Which probability distribution is used when modeling a process with only two possible outcomes, such as success or failure? A. Poisson. B. Binomial. C. Normal. D. Exponential. Answer. The correct answer is B. Binomial. In a normal distribution, what percentage of data falls within two standard deviations of the mean? A. 50%. B. 68%. C. 95%. D. 99.7%. Answer. The correct answer is C. 95%. Which probability distribution has the same value for its mean and variance? A. Binomial. B. Poisson. C. Normal. D. Uniform. Answer. The correct answer is B. Poisson, since lambda. Lambda is both the mean and variance in a Poisson distribution. This detailed guide on probability distributions provides everything you need for your PhD entrance exam in zoology. Let me know the next topic. You said. Sampling distribution. ChatGPT said. Sampling distribution. Introduction to sampling distribution. A sampling distribution is the probability distribution of a given statistic based on a random sample from a population. It describes how a sample statistic, such as mean, proportion, or variance varies from one sample to another when multiple samples are taken from the same population. Understanding sampling distributions is essential in statistical inference, as it forms the basis for hypothesis testing and confidence interval estimation. The key idea is that if we repeatedly take random samples from a population, the values of the sample statistic, such as the mean, will form their own distribution, known as the sampling distribution. Types of sampling distributions. There are different types of sampling distributions depending on the statistic being measured. The most commonly used types include 1. Sampling distribution of the sample mean. When multiple random samples of size n are taken from a population, the means of those samples form a distribution called the sampling distribution of the sample mean. If the population has a mean mu and standard deviation sigma, then the sampling distribution of the sample mean has mean equals mu, same as the population mean, standard deviation equals sigma, n, sigma, called the standard error of the mean, central limit theorem, CLT. If the sample size n is large, typically n is greater than or equal to 30. N is greater than or equal to 30, the sampling distribution of the sample mean will be approximately normal, even if the original population is not normally distributed. 2. Sampling distribution of the sample proportion. If we take repeated samples from a population and calculate the proportion p of individuals with a certain characteristic, these proportions form a sampling distribution. If the population proportion is p, then the sampling distribution of p has mean equals p, standard deviation, standard error equals p 1 minus p n, p 1 minus p. The sampling distribution of the sample proportion is approximately normal if n p is greater than or equal to 5. Np is greater than or equal to 5 and n 1 minus p is greater than or equal to 5. N 1 minus p is greater than or equal to 5, normal approximation condition. 3. Sampling distribution of the sample variance. When we calculate the variance from different random samples, these values form a sampling distribution of the sample variance. If the population follows a normal distribution, then the sample variance follows a chi-square distribution with n minus 1 degrees of freedom. 4. Sampling distribution of the sample standard deviation. The standard deviation calculated from multiple samples forms a sampling distribution. However, it does not follow a normal distribution but a chi-square or related distribution. Properties of sampling distributions. Unbiased estimation. The mean of the sampling distribution of a sample statistic, e.g., mean or proportion equals the true population parameter. Law of large numbers. As sample size increases, the sample mean becomes a better estimate of the population mean. Central limit theorem. Regardless of the population distribution, the sampling distribution of the sample mean tends toward normality as n increases. Standard error measures how much the sample statistic deviates from the true population parameter. Importance of sampling distributions in zoology and biology population studies, estimating mean body length, weight, or behavior patterns in animal populations. Epidemiology, estimating disease prevalence using sample data from a population. Genetics, studying allele frequencies from a subset of a population. Ecology, estimating species richness in an ecosystem from limited sample plots. Example calculation of sampling distribution problem. A researcher is studying the average body length of a fish species in a lake. The population mean body length is 30 centimeters, with a standard deviation of 6 centimeters. If the researcher takes random samples of size 36, what will be the mean and standard deviation of the sampling distribution of the sample mean? Solution, given. Population mean mu equals 30. Mu equals 30 centimeters. Population standard deviation sigma equals 6. Sigma equals 6 centimeters, sample size n equals 36, n equals 36. Using the formula for the standard error of the mean. Standard error equals sigma n equals 6, 36 equals 6, 6 equals 1. Standard error equals n. Sigma equals 36. 6 equals 6, 6 equals 1. Thus, the sampling distribution of the sample mean will have mean equals 30 centimeters, same as the population mean, standard deviation equals 1 centimeter, the standard error. By the central limit theorem, the sampling distribution of the sample mean will be approximately normal. Solved previous year question. According to the central limit theorem, as the sample size n increases, the shape of the sampling distribution of the sample mean A becomes more skewed, B becomes more normal, C becomes bimodal, D becomes uniform. Answer. The correct answer is B, becomes more normal. According to the central limit theorem, regardless of the population's shape, the sampling distribution of the sample mean approaches a normal distribution as the sample size increases. Multiple choice questions, MCQs. Which of the following statements about sampling distributions is true? A. The mean of the sampling distribution is always different from the population mean. B. The standard deviation of the sampling distribution decreases as the sample size increases. C. The sampling distribution is always skewed. D. The sampling distribution follows a Poisson distribution. Answer. The correct answer is B. The standard deviation of the sampling distribution decreases as the sample size increases. The standard deviation of the sampling distribution of the sample mean is called a sampling error. B. Standard deviation. C. Standard error. D. Variance. Answer. The correct answer is C. Standard error. Which of the following conditions ensures that the sampling distribution of the sample proportion is approximately normal? A. The sample size must be less than 5. B. The sample size must be greater than 100. C. NP is greater than or equal to 5. NP is greater than or equal to 5 and N. 1 minus P is greater than or equal to 5. And 1 minus P is greater than or equal to 5. D. The sample proportion must be greater than 0.8. Answer. The correct answer is C. N. P is greater than or equal to 5. NP is greater than or equal to 5 and N. 1 minus P is greater than or equal to 5. And 1 minus P is greater than or equal to 5. This detailed lecture on sampling distributions covers all the theoretical aspects required for the University of Calicut PhD entrance examination in zoology. Let me know the next topic. You said, difference between parametric and non-parametric statistics. ChatGPT said. Difference between parametric and non-parametric statistics. Introduction. Statistical methods are broadly classified into parametric and non-parametric techniques. The choice between them depends on the assumptions about the data distribution, sample size, and the type of variable being analyzed. Understanding these differences is crucial in biological research, particularly in areas like ecology, genetics, and behavioral studies, where data often vary in structure and distribution. Parametric statistics. Parametric tests are statistical methods that make specific assumptions about the underlying population distribution, usually assuming a normal distribution, Gaussian distribution. These tests typically involve parameters such as the mean and variance to describe data. Key characteristics of parametric statistics: assumption of normality. The data should follow a normal distribution, bell-shaped curve. Homogeneity of variance. The variance should be approximately equal across groups. Homoscedasticity. Large sample size preference. Parametric tests work well with larger sample sizes due to the central limit theorem. Continuous data. Parametric tests are applied to continuous or interval ratio data, e.g., height, weight, temperature. More powerful tests. They have greater statistical power when assumptions are met, meaning they are more likely to detect a true effect. Common parametric tests. T-test for comparing two means. ANOVA, analysis of variance, for comparing multiple means. Pearson's correlation coefficient. R, for assessing relationships. Linear regression, for predicting relationships between variables. Example of parametric test in biology. A researcher measures the average weight of two fish species in a lake and wants to determine if there is a significant difference in their weights. Since weight is a continuous variable and follows a normal distribution, a t-test is appropriate. Non-parametric statistics. Non-parametric tests are statistical methods that do not require specific assumptions about the population distribution. These tests are useful when data are skewed, ordinal, categorical, or when the sample size is small. Key characteristics of non-parametric statistics: no assumption of normality. Can be used for data that do not follow a normal distribution. Suitable for small sample sizes, more effective when sample size is too small to assume normality. Ordinal or categorical data. Used for ranked data, e.g., pain scales, survey responses. Handles outliers well. Non-parametric tests are less affected by extreme values compared to parametric tests. Lower statistical power. Less likely to detect a real effect compared to parametric tests when assumptions hold. Common non-parametric tests. Mann-Whitney U-test, equivalent to the t-test for independent samples. Wilcoxon-signed rank test, equivalent to the paired t-test. Kruskal-Wallis test, equivalent to ANOVA. Spearman's rank correlation, for relationships between variables. Chi-square test, for categorical data analysis. Example of non-parametric test in biology. A researcher studies the feeding behavior of different bird species based on a ranking system, e.g., aggressive, moderate, passive. Since the data are ordinal and not normally distributed, the Kruskal-Wallis test would be more appropriate than ANOVA. Key differences between parametric and non-parametric statistics. Assumptions about distribution. Parametric tests assume a normal distribution. Non-parametric tests do not require normality. Type of data used. Parametric tests: continuous, interval, or ratio data. Non-parametric tests: ordinal, ranked, or categorical data. Handling of outliers. Parametric tests are sensitive to outliers. Non-parametric tests are robust against extreme values. Statistical power. Parametric tests are more powerful when assumptions are met. Non-parametric tests are less powerful but more flexible. Application in biology. Parametric tests are used when analyzing body weight, enzyme activity, blood pressure, etc. Non-parametric tests are used for animal behavior studies, ecological ranking, and genetic data analysis. Solved previous year question. Question. Which of the following situations is best suited for using a non-parametric test instead of a parametric test? A. Comparing the means of two normally distributed datasets with equal variances. B. Analyzing survey data where responses are recorded as poor, average, good, excellent. C. Studying the correlation between two continuous, normally distributed variables. D. Testing the effectiveness of a new drug where sample size is large and data is normally distributed. Answer. The correct answer is B. Analyzing survey data where responses are recorded as poor, average, good, excellent. Since survey responses are ordinal, ranked rather than continuous, a non-parametric test like the Kruskal-Wallis test is more appropriate. Multiple choice questions, MCQs. Which of the following is an assumption of parametric statistical tests? A. Data must be ranked. B. Data must be normally distributed. C. Sample size must be less than 10. D. Data should be categorical. Answer. B. Data must be normally distributed. Which statistical test is appropriate for comparing the medians of two independent groups when the data is skewed? A. T-test. B. ANOVA. C. Mann-Whitney U-test. D. Pearson's correlation. Answer. C. Mann-Whitney U-test. Which non-parametric test is used as an alternative to the paired t-test? A. Wilcoxon signed rank test. B. Chi-square test. C. Kruskal-Wallis test. D. Spearman's rank correlation. Answer: A. Wilcoxon signed rank test. This detailed lecture provides a comprehensive understanding of parametric and non-parametric statistics, covering theory, applications, previous year question solutions, and MCQs. Let me know the next topic for your University of Calicut PhD entrance exam in zoology preparation. You said. Confidence interval. ChatGPT said. Confidence interval introduction. A confidence interval, C, is a statistical tool used to estimate a population parameter, such as the mean or proportion based on a sample. It provides a range of values that is likely to contain the true population parameter with a certain level of confidence. For example, if a 95% confidence interval for the mean body length of a fish species is 25 centimeters, 30 centimeters, it means that we are 95% confident that the true mean body length of all fish in that species falls within this range. Confidence intervals are widely used in biological research, including population studies, ecological surveys, genetics, and medical research, to make informed predictions about biological phenomena. Concept of confidence level. The confidence level represents the probability that the confidence interval contains the true population parameter. It is usually expressed as 90%, 95%, or 99%. A 95% confidence level means that if we were to take 100 different samples, approximately 95 of them would contain the true population parameter. A 99% confidence level provides a wider interval, meaning we are more confident but with less precision. The most commonly used confidence level is 95%, as it provides a balance between accuracy and precision. Formula for confidence interval. The general formula for a confidence interval for a population mean mu. Mu is Ci equals x plus or minus c times sigma n. C equals x plus or minus c times n. Sigma, where x equals sample mean z. C equals z score depends on confidence level sigma. Sigma equals population standard deviation n. N equals sample size. If the population standard deviation sigma is unknown, we use the sample standard deviation s. S and replace the z score with a t-score from the t-distribution. For proportions, the confidence interval is calculated as Ci equals p caret plus or minus c times p caret 1 minus p caret n. C equals p caret plus or minus c times n. P caret 1 minus p caret, where p caret equals sample proportion z. C equals z score based on confidence level n. N equals sample size. Scores for common confidence levels. Confidence level z score 90% 1.645. 95% 1.96, 99% 2.576. The higher the confidence level, the wider the confidence interval, meaning we are more confident, but the estimate becomes less precise. Interpretation of confidence interval. Correct interpretation. A 95% confidence interval for the average weight of a species of frog is 20 grams, 25 grams. This means that if we take 100 different random samples, 95 out of 100 will have the true mean weight within this range. Incorrect interpretation. There is a 95% probability that the true mean weight is between 20 grams and 25 grams. Incorrect, because the true mean is a fixed value, not a probability. Factors affecting confidence interval. Sample size n. Larger samples lead to narrower confidence intervals, more precise estimates. Smaller samples result in wider confidence intervals, less precision. Confidence level. Higher confidence levels 99% give wider intervals. Lower confidence levels 90% give narrower intervals. Variability in data. Larger samples lead to narrower confidence intervals, more precise estimates. Smaller samples result in wider confidence intervals, less precision. Confidence level. Higher confidence levels 99% give wider intervals. Lower confidence levels 90% give narrower intervals. Variability in data. Standard deviation. Higher variability, larger standard deviation, results in a wider confidence interval. Lower variability leads to a narrower interval. Example calculation of confidence interval problem. A researcher measures the average blood pressure of 50 monkeys in a population. The sample mean blood pressure is 120 mm of mercury, and the sample standard deviation is 15 mm of mercury. Compute the 95% confidence interval for the true mean blood pressure of monkeys in the population. Solution, given x equals 120, x equals 120 mm of mercury, sample mean s equals 15. S equals 15 mm of mercury, sample standard deviation n equals 50. N equals 50, sample size z equals 1.96. Z equals 1.96 for 95% confidence level. Step 1, compute the standard error SE. SE equals SN equals 15, 50 equals 15, 7.07 equals 2.12. SE equals N. S equals 50. 15 equals 7.07, 15 equals 2.12. Step 2, compute the margin of error ME. ME equals Z times SE equals 1.96 times 2.12 equals 4.15. Me equals Z times SE equals 1.96 times 2.12 equals 4.15. Step 3, compute the confidence interval CI. CI equals x plus or minus ME equals 120 plus or minus 4.15. C equals x plus or minus me equals 120 plus or minus 4.15. C. I equals 115.85. 124.15. C equals 115.85. 124.15. Final answer. The 95% confidence interval for the mean blood pressure of monkeys is 115.85 mm of mercury, 124.15 mm of mercury. Solved previous year question. A researcher calculates a 95% confidence interval for the mean lifespan of a species of birds and finds it to be 8.4 years, 10.2 years. What does this mean? A. There is a 95% probability that the lifespan of a randomly selected bird is between 8.4 and 10.2 years. B. The true mean lifespan of the species is definitely between 8.4 and 10.2 years. C. If we take multiple samples, 95% of them will give an interval that contains the true mean lifespan. D. The sample mean must be exactly 9.3 years. Answer. The correct answer is C. If we take multiple samples, 95% of them will give an interval that contains the true mean lifespan. Explanation. Confidence intervals refer to the long-term proportion of intervals that contain the true population mean, not to the probability of a specific interval containing the mean. Multiple choice questions MCQs. Which factor decreases the width of a confidence interval? A. Increasing the confidence level. B. Increasing the sample size. C. Increasing the standard deviation. D. Decreasing the sample size. Answer. B. Increasing the sample size. A wider confidence interval suggests more precision. B. Less variability in data. C. More uncertainty in the estimate. D. A smaller sample size is used. Answer. C. More uncertainty in the estimate. If a researcher wants a more precise estimate, they should use a higher confidence level. B. Increase the sample size. C. Use a lower standard deviation. D. Decrease the mean. Answer. B. Increase the sample size. This comprehensive guide on confidence intervals covers theory, formulas, examples, previous year solved questions, and MCQs, making it an essential study material for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, errors, ChatGPT said. Errors in statistical analysis introduction. Errors in statistical analysis refer to deviations from the true value due to limitations in data collection, sampling, or measurement. These errors can significantly impact the accuracy of results, leading to incorrect conclusions. In biological research, errors can arise in experimental design, data collection, and statistical interpretation, making it crucial to understand their types, sources, and implications. Errors in statistics are broadly classified into systematic errors and random errors. Additionally, errors related to hypothesis testing, such as type 1 and type 2 errors, play a crucial role in decision-making. Types of errors. 1. Systematic errors. Bias. Systematic errors occur due to flaws in the experimental setup, data collection process, or observer bias. These errors lead to consistent deviations from the true value, making results inaccurate but reproducible. Causes of systematic errors. Instrumental errors, faulty or improperly calibrated instruments, e.g., a misaligned spectrophotometer in molecular biology. Observer bias, subjective errors made by researchers, e.g., miscounting cells under a microscope. Environmental factors, uncontrolled variables like temperature, humidity, or pH affecting results. Selection bias, non-random selection of subjects, leading to skewed results, e.g., selecting only healthy individuals in a disease study. Example of systematic error. In a study measuring oxygen consumption in fish, if the oxygen sensor is calibrated incorrectly, all measurements will be consistently higher or lower than the true value. Solution. Use proper calibration techniques for instruments. Apply randomization and blinding to reduce observer bias. Standardize environmental conditions during data collection. 2. Random errors. Random errors are unpredictable variations that occur due to chance fluctuations in measurement or sampling. Unlike systematic errors, random errors do not show a consistent pattern and tend to average out over multiple trials. Causes of random errors. Biological variability, natural differences among individuals in a population, measurement fluctuations, inconsistencies in instrument sensitivity, sampling errors, differences arising due to the use of a subset of the population. Example of random error. If multiple researchers weigh the same specimen on a digital balance, slight variations occur due to fluctuations in electronic measurement. Solution. Increase sample size to minimize random errors. Repeat experiments multiple times and take the average value. Use high-precision instruments to reduce variability. Errors in hypothesis testing. 1. Type 1 error, false positive. A type 1 error occurs when a true null hypothesis is incorrectly rejected. This means that the study concludes an effect exists when, in reality, it does not. Example. A new anti-cancer drug is tested, and the results indicate it is effective when it actually has no effect. Probability of type 1 error. The probability of committing a type 1 error is represented by alpha, typically set at 0.055% in biological studies. Solution. Lower alpha level, e.g., using 0.01 instead of 0.05 reduces the risk of type 1 error but increases type 2 error. Use larger sample sizes for better statistical power. 2. Type 2 error, false negative. A type 2 error occurs when a false null hypothesis is not rejected. This means that the study fails to detect an effect that truly exists. Example. A researcher studies the effect of a pesticide on honeybee mortality but concludes that the pesticide has no effect, when in reality, it does harm the bees. Probability of type 2 error. The probability of committing a type 2 error is represented by beta, and 1 beta is statistical power, which indicates the likelihood of correctly detecting an effect. Solution. Increase sample size to improve detection power. Use a higher alpha, e.g., 0.1 instead of 0.05 when detecting subtle effects is more critical. Improve experimental design to reduce noise and variability. Statistical power and error relationship. There is a trade-off between type 1 and type 2 errors. Reducing alpha, type 1 error, increases beta, type 2 error, making it harder to detect true effects. Increasing alpha reduces beta but raises the risk of false positives. Thus, researchers must balance the risk of both errors based on the biological and experimental context. Example calculation of error probability problem. A researcher is testing a new drug for malaria. The null hypothesis states that the drug has no effect. The researcher sets alpha equals 0.05 and conducts the experiment. If the drug actually has no effect, but the researcher rejects the null hypothesis, what type of error is this? If the drug truly reduces malaria cases, but the researcher fails to reject the null hypothesis, what type of error is this? Solution. Rejecting a true null hypothesis equals type 1 error, false positive. Failing to reject a false null hypothesis equals type 2 error, false negative. Thus, the first scenario leads to incorrectly claiming that the drug is effective, while the second scenario results in missing out on a truly beneficial drug. Solved previous year question. A researcher is conducting a genetic study and sets the significance level at 0.01 instead of 0.05. What effect does this have? A. Increases the risk of type 1 error. B. Decreases the risk of type 1 error but increases type 2 error. C. Increases both type 1 and type 2 errors. D. Has no effect on the errors. Answer. The correct answer is B. Decreases the risk of type 1 error but increases type 2 error. Explanation. Lowering the significance level reduces the chance of falsely rejecting the null hypothesis, but it also increases the likelihood of missing a real effect, type 2 error. Multiple choice questions, MCQs. Which of the following is an example of a systematic error? A. Variability in data due to sampling. B. An instrument consistently giving readings 2 degrees Celsius higher than the true value. C. Random fluctuations in experimental conditions. D. Measurement errors that cancel out over multiple trials. Answer. B. An instrument consistently giving readings 2 degrees Celsius higher than the true value. If a researcher incorrectly concludes that a new drug is effective when it is not, this is an example of a type 1 error. B. Type 2 error. C. Random error. D. Systematic error. Answer. A type 1 error. Which of the following decreases the probability of a type 2 error? A. Decreasing sample size. B. Increasing sample size. C. Lowering the significance level. D. Increasing random error. Answer. B. Increasing sample size. Conclusion. Understanding errors in statistics is crucial for accurate biological research and hypothesis testing. Systematic errors bias must be minimized through proper calibration and experimental design, while random errors can be reduced by increasing sample size. Additionally, the trade-off between type 1 and type 2 errors should be carefully balanced to ensure meaningful and reliable conclusions in zoological research. This detailed guide provides theoretical concepts, examples, solved previous year questions, and MCQs, making it a comprehensive resource for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Levels of significance. ChatGPT said. Levels of significance in statistical analysis. Introduction. The level of significance, alpha, is a fundamental concept in hypothesis testing that determines the probability of rejecting a true null hypothesis. It represents the threshold beyond which we consider an observed result statistically significant. In biological research, setting an appropriate significance level is critical to avoid incorrect conclusions. The level of significance is typically set at 0.05, 5%, but 0.01, 1%, or even 0.1, 10% may be used depending on the study's nature and the consequences of errors. Understanding levels of significance helps in making informed decisions in zoology, ecology, and biomedical sciences, where statistical analysis plays a key role in validating experimental results. Definition of level of significance, alpha. The level of significance, alpha, is the probability of making a type 1 error, rejecting a true null hypothesis. It quantifies the likelihood of observing a result by chance alone rather than due to a real effect. Common significance levels: 0.05, 5%, right pointing arrow most commonly used in biological studies. 0.01, 1%, right pointing arrow used in critical medical or genetic research where errors must be minimized. 0.10, 10%, right pointing arrow sometimes used in exploratory studies where missing a potential effect is more critical than making a false claim. For example, if a researcher sets alpha equals 0.05, it means there is a 5% probability that the observed result is due to chance alone, and 95% confidence that it is due to an actual effect. Relationship between alpha and hypothesis testing. Hypothesis testing involves comparing an observed statistic with a critical value determined by alpha. If p is less than or equal to alpha, we reject the null hypothesis H and conclude that the observed effect is statistically significant. If p and gt, alpha, we fail to reject the null hypothesis H and conclude that there is insufficient evidence to support the alternative hypothesis. Example in biological research. A researcher studying the effect of a pesticide on insect mortality sets alpha equals 0.05. If a statistical test yields a p-value of 0.03, which is less than 0.05, the researcher rejects H and concludes that the pesticide has a significant effect on mortality. Choosing an appropriate level of significance. The choice of alpha depends on field of study. In medical research, where incorrect conclusions can have serious consequences, alpha equals 0.01 or lower is often used. In ecological or behavioral studies, where errors may not have life-threatening consequences, alpha equals 0.05 or 0.1 might be used. Risk of type 1 versus type 2 errors. Lower alpha, e.g., 0.01, reduces the probability of type 1 error, false positives, but increases the risk of type 2 error, false negatives. Higher alpha, e.g., 0.10, reduces the risk of missing true effects but increases false positives. Example. A genetic study testing for a rare mutation might set alpha equals 0.01 to avoid false positives. A preliminary ecological survey might use alpha equals 0.1 to ensure no potential effects are overlooked. Critical value and level of significance. The critical value is the threshold that determines statistical significance. It is based on alpha and the probability distribution of the test statistic. E.g., normal distribution, t-distribution. For a two-tailed test with alpha equals 0.05, the critical regions are the extreme 2.5% on both ends of the normal distribution. If the test statistic falls inside this region, H is rejected. If the test statistic falls outside, H is not rejected. For a one-tailed test, the entire alpha is on one side, making it more powerful for detecting effects in a specific direction. Impact of changing alpha in hypothesis testing. Lowering alpha, e.g., from 0.05 to 0.01. Reduces false positives, type 1 error. Requires larger sample sizes for sufficient power. May increase type 2 error, false negatives. Increasing alpha, e.g., from 0.05 to 0.1. Increases the chance of detecting real effects. Raises the risk of false positives. Example of impact on a zoological study. A study on fish population decline due to pollution uses alpha equals 0.05. If researchers lower alpha to 0.01, they reduce the risk of falsely concluding pollution is responsible. However, this increases the chance of missing a real effect, higher type 2 error, leading to inaction. Solved previous year question. A researcher is testing a new antibiotic's effectiveness and sets a significance level at 0.01 instead of 0.05. What does this mean? A. It increases the risk of type 1 error. B. It decreases the risk of type 1 error but increases type 2 error. C. It increases both type 1 and type 2 errors. D. It has no effect on the errors. Answer. B. It decreases the risk of type 1 error but increases type 2 error. Explanation. Lowering alpha from 0.05 to 0.01 makes it harder to falsely conclude that the antibiotic is effective reducing type 1 error. However, it also makes it harder to detect a true effect, increasing the likelihood of type 2 error. Multiple choice questions. MCQs. If a researcher sets alpha equals 0.01 instead of 0.05, what is the effect? A. Increases the likelihood of rejecting HB, decreases the probability of type 1 error. C. Increases the probability of type 1 error. D. Reduces the risk of type 2 error. Answer. B. Decreases the probability of type 1 error. A significance level of 0.05 means there is a 5% probability of committing a type 2 error. B. There is a 5% probability that the null hypothesis is true. C. There is a 5% chance of rejecting a true null hypothesis. D. There is a 5% probability of accepting a false null hypothesis. Answer. C. There is a 5% chance of rejecting a true null hypothesis. Which of the following factors should influence the choice of alpha? A. The consequences of making a type 1 error. B. The consequences of making a type 2 error. C. The field of study and research objective. D. All of the above. Conclusion. Level of significance alpha is a crucial aspect of hypothesis testing that determines the probability of type 1 error. Choosing an appropriate alpha level depends on the research context, risk of errors, and the importance of findings. Lower alpha values increase the reliability of results but make it harder to detect real effects, while higher alpha values increase sensitivity but risk more false positives. This detailed guide provides theoretical concepts, examples, solved previous year questions, and MCQs, making it a complete resource for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Assumptions and techniques of ANOVA. ChatGPT said. Analysis of variance. ANOVA. Assumptions and techniques. Introduction. Analysis of variance. ANOVA is a statistical method used to compare the means of multiple groups to determine if there is a statistically significant difference between them. It is widely used in biological research, including ecology, genetics, physiology, and pharmacology, to analyze experimental data where multiple conditions or treatments are compared. ANOVA extends the t-test, which compares only two groups, to more than two groups while controlling the overall error rate. The key concept behind ANOVA is to partition the total variance in a dataset into variance due to different treatments, between group variance, and variance due to random error within group variance. If the between group variance is significantly larger than the within group variance, we conclude that at least one group mean is different from the others. Assumptions of ANOVA. For ANOVA to produce valid results, certain assumptions must be met. 1. Independence of observations. Each sample must be independent, meaning that observations in one group should not influence observations in another. This assumption ensures that measurements are not correlated. Example, in a study on the effect of different fertilizers on plant growth, each plant should be independent, and measurements should not be affected by neighboring plants. 2. Normality of data. The data within each group should follow a normal distribution. This assumption is crucial for small sample sizes but becomes less critical for larger datasets due to the central limit theorem, CLT. Example, if a study examines blood pressure differences among 3 diet groups, each group's blood pressure readings should ideally follow a normal distribution. If a study examines blood pressure differences among 3 diet groups, each group's blood pressure readings should ideally follow a normal distribution. Checking normality. Graphical methods, histograms, QQ plots, statistical tests, Shapiro-Wilk test, Kolmogorov-Smirnov test. 3. Homogeneity of variance, homoscedasticity. The variance within each group should be approximately equal. If variances differ significantly, the results of ANOVA may be biased. Example, if different diets affect weight gain in rats, the variation in weight gain across all diet groups should be similar. Checking homogeneity. Levine's test common for biological data. Bartlett's test assumes normality. 4. Continuous and independent dependent variable. The dependent variable should be continuous, e.g., height, weight, enzyme activity, and measured on an interval or ratio scale. Example, if comparing react times of different insect species, reaction time must be measured in seconds rather than categories. 5. Balanced design preferable, but not strictly necessary. Although ANOVA can handle unequal sample sizes, it works best when each group has an equal number of observations to maintain statistical power. Types of ANOVA and their techniques. 1. One-way ANOVA, single-factor ANOVA. Used when comparing means across one independent variable, factor, with multiple levels. Example, a zoologist wants to compare the average lifespan of 3 species of butterflies. The independent variable is species, and the dependent variable is lifespan. Steps for one-way ANOVA. Formulate null H and alternative H hypotheses. H, all group means are equal. H, at least one group mean is different. Calculate the F ratio, which is F equals between group variance within group variance. F equals within group variance between group variance. Compare the calculated F value with the critical F value from an F distribution table at a chosen significance level, alpha equals 0.05. If F and GT, critical value, reject H and conclude that at least one group differs. Post hoc tests if significant differences are found. 2P's HSD for pairwise comparisons. Bonferroni correction adjusts p-values for multiple comparisons. 2. Two-way ANOVA, factorial ANOVA. Used when examining the effect of 2 independent variables on a single dependent variable. Example, a biologist tests how temperature, low, medium, high, and humidity, low, high, affect insect metabolism. Key feature. Tests main effects. Each factors impact separately. Tests interaction effects, how factors work together. Interpretation. A significant main effect means one factor influences the outcome. A significant interaction means the effect of one factor depends on another factor. 3. Repeated measures ANOVA. Used when the same subjects are measured multiple times under different conditions, e.g., before, during, and after treatment. Example, studying heart rate changes in frogs exposed to different water temperatures over time. Advantage, controls for individual variability, increasing statistical power. Challenge, requires sphericity, equal variances of differences tested using Mauchly's test. 4. ANOVA multivariate analysis of variance extends ANOVA when multiple dependent variables are analyzed simultaneously. Example, a physiologist studies oxygen consumption and heart rate in different mammal species under varying temperatures. Technique, uses Wilks lambda instead of F test. Solved previous year question. Which of the following assumptions is necessary for conducting ANOVA? A. The samples must have different means. B. The samples must be dependent on each other. C. The populations must have equal variances. D. The population distributions must be skewed. Answer. C. The populations must have equal variances. Explanation. Homogeneity of variance is a key assumption in ANOVA, ensuring that differences among groups are due to the independent variable rather than unequal variability. Multiple choice questions, MCQs. Which of the following statements is true about ANOVA? A. It compares variances instead of means. B. It requires normally distributed data. C. It is used only for 2 groups. D. It does not require independence of observations. Answer. B. It requires normally distributed data. What does the F statistic in ANOVA represent? A. The ratio of total variance to within group variance. B. The ratio of between group variance to within group variance. C. The ratio of mean variance to standard deviation. D. The ratio of sum of squares to degrees of freedom. Answer. B. The ratio of between group variance to within group variance. What is the purpose of a post hoc test in ANOVA? A. To determine whether the data meets normality assumptions. B. To compare individual group means after finding a significant F-test. C. To calculate the test statistic before conducting ANOVA. D. To adjust the degrees of freedom. Answer. B. To compare individual group means after finding a significant F-test. Conclusion. ANOVA is a powerful technique for comparing multiple group means while controlling error rates. It relies on assumptions of independence, normality, and homogeneity of variance to produce valid results. The F-ratio is used to determine statistical significance, and post hoc tests help identify which groups differ. This guide provides a detailed theory explanation, assumptions, techniques, solved previous year question, and MCQs, making it a complete package for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, one-way and two-way classification. ChatGPT said. One-way and two-way classification in statistics. Introduction. Classification in statistics refers to the process of organizing data based on one or more criteria factors. In experimental and observational studies, data are often categorized using one-way classification, single-factor classification, and two-way classification, double-factor classification. These classifications are essential for designing experiments, analyzing variance, ANOVA, and understanding interactions between variables in biological and zoological studies. One-way classification definition. One-way classification occurs when observations are classified based on a single factor or characteristic. It is commonly used in one-way ANOVA, where data is grouped based on one independent variable with multiple levels categories. Example in biology. Suppose a zoologist studies the effect of different diets on the body weight of fish. The independent variable factor is diet type, with 3 levels. Diet A, Diet B, Diet C. Each fish is assigned to one of these 3 diet groups, and weight gain is recorded. The goal is to determine whether diet type significantly affects weight gain. Mathematical representation. If there are K groups, the observation X IJ represents the J TH observation in the I TH group. The total variation in the dataset is split into total variation equals between group variation plus within group variation. Total variation equals between group variation plus within group variation, where between group variation measures differences in means across groups. Within group variation accounts for random variation within each group. The F statistic is calculated as F equals mean square between groups MSB, mean square within groups MSW. F equals mean square within groups MSW, mean square between groups MSB. A significant F value implies that at least one group mean is different from the others. Advantages of one-way classification: simpler and easier to analyze. Helps compare the effect of a single factor on the dependent variable. Useful in controlled biological experiments, e.g., effect of temperature on enzyme activity. Limitations. Ignores interactions with other potential influencing factors. Cannot analyze multiple factors simultaneously. Two-way classification. Definition. Two-way classification occurs when observations are classified based on 2 factors, independent variables. It is used in two-way ANOVA, where 2 categorical variables influence the dependent variable. Example in biology. A biologist studies how temperature and humidity affect insect metabolism. There are factor 1 temperature low, medium, high, factor 2 humidity low, high. Each insect is assigned to one combination of temperature and humidity, and metabolic rate is recorded. The goal is to determine if temperature affects metabolism, if humidity affects metabolism, if temperature and humidity interact to influence metabolism. Mathematical representation. For each observation X IJK I represents levels of factor A, e.g., temperature. J represents levels of factor B, e.g., humidity. K represents replicates within each combination. The total variance is divided into total variation equals variation due to factor A plus variation due to factor B plus interaction effect plus error. Total variation equals variation due to factor A plus variation due to factor B plus interaction effect plus error. Factor A, main effect of the first independent variable, e.g., temperature. Factor B, main effect of the second independent variable, e.g., humidity. Interaction effect A times B determines if the effect of one factor depends on the level of the other. Interpretation of two-way ANOVA. If factor A is significant, it means temperature significantly affects metabolism. If factor B is significant, it means humidity significantly affects metabolism. If the interaction effect is significant, it means temperature and humidity work together in influencing metabolism. Advantages of two-way classification. Allows analysis of multiple factors simultaneously. Identifies interaction effects, which are essential in biological systems. Increases statistical efficiency by reducing random error. Limitations. More complex than one-way classification. Requires larger sample sizes to detect significant effects. Assumes normality and homogeneity of variance. Comparison of one-way and two-way classification. Feature one-way classification, two-way classification. Number of factors 1, 2. Example: effect of diet on weight gain, effect of temperature and humidity on metabolism. Hypotheses: tests if one factor influences the dependent variable. Tests if each factor and their interaction affect the dependent variable. ANOVA used one-way ANOVA, two-way ANOVA. Complexity: simple, more complex interactions considered. No, yes, solved previous year question. A researcher wants to study the effect of 2 different fertilizers, A and B, on plant growth. He also considers 2 different soil types, clay and sandy. Which statistical test should be used? A. One-way ANOVA. B. Two-way ANOVA. C. Chi-square test. D. Regression analysis. Answer. B. Two-way ANOVA. Explanation: since there are 2 factors, fertilizer type and soil type, a two-way ANOVA is appropriate to analyze the main effects and interaction effects between these 2 factors. Multiple choice questions, MCQs. In one-way classification, data is categorized based on a. 1 independent variable. B. 2 independent variables. C. 3 independent variables. D. No independent variable. Answer: a. 1 independent variable. What additional information does two-way classification provide compared to one-way classification? A. Whether sample sizes are equal. B. Whether the dependent variable is continuous. C. Whether the two factors interact with each other. D. Whether variance is normally distributed. Answer. C. Whether the two factors interact with each other. Which assumption is common to both one-way and two-way ANOVA? A. The dependent variable must be categorical. B. The groups must have different sample sizes. C. The population variances must be equal. D. The independent variables must be correlated. Answer. C. The population variances must be equal. Conclusion. One-way and two-way classification are fundamental concepts in statistical analysis, particularly in biological research. One-way classification deals with a single independent variable, while two-way classification involves two independent variables and their interaction effects. One-way ANOVA is used when analyzing a single factor, whereas two-way ANOVA is used when studying two factors and their combined effects. This detailed guide provides comprehensive theory, solved previous year question, and MCQs to ensure complete preparation for the University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. No table format representation, please. ChatGPT said. One-way and two-way classification in statistics introduction. In experimental and observational studies, data are often classified based on one or more factors, independent variables. These classifications are vital for understanding how different factors influence the dependent variable, especially when designing experiments in biological and zoological research. The two major types of classification are one-way classification and two-way classification. One-way classification definition. One-way classification refers to a scenario where observations are grouped based on a single factor or characteristic. It is commonly used in one-way ANOVA, where data is classified according to a single independent variable with multiple levels or categories. Example in biology. Suppose a zoologist is studying the effect of different diets on the body weight of fish. The independent variable, in this case, is the type of diet, which has three levels: diet A, diet B, and diet C. The zoologist would observe how each diet influences weight gain in fish. The goal of this experiment is to determine whether different diets lead to significant differences in body weight. Mathematical representation for data classified into K. K groups, each observation X. IJXIJ. Represents the J. THJTH. Observation within the I. THITH. Group, the total variation in the data is partitioned into two components. Between group variation, which reflects the differences in means across groups. Within group variation, which reflects the variation within each group. The F statistic is calculated as the ratio of the mean square between groups, MSB, to the mean square within groups, MSW. F equals MSB MSW F equals MSW MSB. If the F value is significant, it suggests that at least one group mean is different from the others. Advantages of one-way classification. It is simpler and easier to analyze, particularly for controlled experiments with a single independent variable. It is useful in biological studies where the effects of one factor, e.g., temperature, diet, or drug, on a dependent variable are being studied. Limitations. It does not account for interactions with other potential influencing factors. It can only analyze the effects of one factor at a time, missing the potential combined effects of multiple variables. Two-way classification. Definition. Two-way classification occurs when observations are classified based on two factors, independent variables. This classification is analyzed using two-way ANOVA, which allows researchers to study how two factors influence a dependent variable both individually and in combination. Example in biology. A biologist is studying how temperature and humidity affect the metabolic rate of insects. In this case, temperature low, medium, high, and humidity low, high are the two factors. Each insect is subjected to one of the 6 possible combinations of temperature and humidity. The goal is to determine if temperature influences metabolic rate, if humidity influences metabolic rate, if temperature and humidity interact to affect metabolic rate. Mathematical representation. For each observation X. IJKXIJK. Where I represents levels of the first factor, temperature, J. J represents levels of the second factor, humidity, and K. K represents replicates within each combination. The total variation is partitioned into variation due to factor A, main effect of temperature, variation due to factor B, main effect of humidity. Interaction effect A times B, which captures whether the effect of one factor depends on the level of the other, error variation. Interpretation of two-way ANOVA. If factor A, temperature, is significant, it means temperature has a significant effect on metabolic rate. If factor B, humidity, is significant, it means humidity significantly influences metabolic rate. If the interaction effect A times B is significant, it means the impact of temperature on metabolic rate depends on the level of humidity, and vice versa. Advantages of two-way classification. It allows analysis of two factors simultaneously, which is more efficient than analyzing them separately. It helps in understanding the interaction between factors, which is crucial in biological systems where variables do not operate in isolation. It increases statistical efficiency by reducing random error, as it accounts for more information from the same set of data. Limitations. Two-way classification is more complex than one-way classification and requires a larger sample size to detect significant effects. Assumptions such as normality and homogeneity of variance must be met, and these assumptions can be more challenging when dealing with multiple factors. Solved previous year question. Question. A researcher wants to study the effect of two different fertilizers, A and B, on plant growth. He also considers two different soil types, clay and sandy. Which statistical test should be used? A. One-way ANOVA. B. Two-way ANOVA. C. Chi-square test. D. Regression analysis. Answer. B. Two-way ANOVA. Explanation. The study involves two factors, fertilizer type and soil type, and the goal is to assess both the individual effects of each factor and their interaction. Hence, two-way ANOVA is appropriate for this experiment. Multiple choice questions, MCQs, in one-way classification, data is categorized based on A. One independent variable. B. Two independent variables. C. Three independent variables. D. No independent variable. Answer. One independent variable. What additional information does two-way classification provide compared to one-way classification? A. Whether sample sizes are equal. B. Whether the dependent variable is continuous. C. Whether the two factors interact with each other. D. Whether variance is normally distributed. Answer. C. Whether the two factors interact with each other. Which assumption is common to both one-way and two-way ANOVA? A. The dependent variable must be categorical. B. The groups must have different sample sizes. C. The population variances must be equal. D. The independent variables must be correlated. Answer. C. The population variances must be equal. Conclusion. One-way and two-way classification are fundamental concepts in statistical analysis, particularly in biological and zoological research. One-way classification deals with a single independent variable, whereas two-way classification analyzes two independent variables and their interactions. Understanding these classifications is crucial for properly designing experiments, analyzing variance, and interpreting the results of biological studies. This guide provides a detailed overview, a solved example, and multiple-choice questions to aid in preparation for the University of Calicut PhD entrance exam in zoology. Let me know the next topic you'd like to cover. You said. Basic Introduction to Multivariate Statistics, ChatGPT said. Basic Introduction to Multivariate Statistics Overview. Multivariate statistics is a branch of statistics that deals with the simultaneous analysis of multiple variables. Unlike univariate statistics, which focuses on a single variable, multivariate statistics is concerned with datasets that involve multiple interrelated variables. The primary aim of multivariate analysis is to understand the relationships among the variables and to summarize the data in ways that allow for the identification of patterns, trends, or classifications within complex data. Multivariate statistics is particularly important in fields like biology, medicine, economics, and social sciences, where multiple variables interact with each other. In biological research, for example, the effects of several different environmental factors, such as temperature, humidity, and light intensity on an organism's growth can be better understood using multivariate statistical methods. Key concepts in multivariate statistics. 1. Multivariate data. Multivariate data refers to data that includes more than one variable, where the number of observations is typically larger than the number of variables. For instance, in a study of the growth rates of different species of animals across various environmental factors, measurements could include variables such as body weight, temperature, humidity, and food intake. The challenge in analyzing such data lies in the fact that these variables may not be independent, but rather may be correlated. 2. Multivariate analysis techniques. Several techniques are employed in multivariate statistics to analyze data. The most common ones include principal component analysis (PCA). This technique reduces the dimensionality of the data by transforming the original variables into a smaller number of new variables, known as principal components, which explain most of the variance in the data. PCA is often used for exploratory data analysis, pattern recognition, and data visualization. Factor analysis. This method is used to identify the underlying relationships between variables by grouping them into factors. The goal is to reduce the number of variables in a dataset by identifying a smaller number of latent factors that explain the variance in the observed data. Multivariate analysis of variance. MANOVA. MANOVA is an extension of ANOVA, analysis of variance, that deals with more than one dependent variable simultaneously. It tests whether there are any statistically significant differences between the means of different groups on a combination of dependent variables. Cluster analysis. This technique classifies objects into groups or clusters based on similarities in their multivariate measurements. The goal is to identify groups that are internally homogeneous but different from other groups. Discriminant analysis. This method is used for classifying observations into predefined categories based on their characteristics. It is often used in fields such as ecology and medicine for classification purposes, e.g., classifying patients based on their symptoms. Canonical correlation analysis (CCA). This technique assesses the relationship between two sets of multivariate variables. For example, it could be used to understand how a set of environmental variables relates to a set of biological measurements. Applications of multivariate statistics. 1. Biological sciences. In biology, multivariate statistics is essential for analyzing complex biological data, such as ecological studies where researchers analyze the relationship between multiple environmental variables and the diversity of species. Genomics to study gene expression patterns across multiple conditions or species. Behavioral studies involving the analysis of several behavioral traits to understand patterns of animal behavior. 2. Medical and clinical research. Multivariate statistics is frequently applied in medical fields to analyze the relationships between various physiological parameters, such as identifying risk factors for diseases by examining multiple medical test results simultaneously. Analyzing the effects of several treatments on patients' health outcomes. 3. Market research and social sciences. Multivariate analysis is widely used in market research to understand consumer preferences based on multiple factors such as age, income, education, and buying behavior. In the social sciences, multivariate methods are used to analyze relationships between multiple demographic, social, and psychological factors. Advantages of multivariate statistics. Simultaneous analysis of multiple variables. Multivariate techniques allow researchers to analyze multiple variables at the same time, which is often necessary in biological, medical, and social research where multiple factors interact. Reduction of data complexity. Techniques like PCA reduce the dimensionality of large datasets, making them easier to interpret and visualize. Improved predictive power. Multivariate methods allow for better prediction models, as they take into account the relationships between variables rather than treating them in isolation. Handling correlated variables. Multivariate statistics help deal with the multicollinearity problem, i.e., when variables are correlated with each other, which would be difficult to handle using univariate statistics. Challenges of multivariate statistics. Complexity and interpretation. Multivariate statistical models are more complex and require careful interpretation, particularly when dealing with a large number of variables. Assumptions of normality and homogeneity. Many multivariate methods assume that the data are normally distributed and that variances and covariances are homogeneous, which may not always hold true in real-world datasets. Large sample sizes. To achieve reliable results, multivariate methods often require large sample sizes, which can be difficult to obtain, especially in specialized biological studies. Solved previous year question. Question. Which of the following is most appropriate for reducing the dimensionality of a dataset in which there are many correlated variables? A. Principal component analysis (PCA). B. Analysis of variance ANOVA. C. Cluster analysis. D. Linear regression. Answer. A. Principal component analysis (PCA). Explanation. PCA is the most suitable method for reducing the dimensionality of a dataset with many correlated variables. It identifies the principal components, which are new variables that capture most of the variance in the data. PCA helps simplify the analysis and visualization of high-dimensional data by transforming the original variables into fewer uncorrelated components. Multiple choice questions (MCQs). Which of the following techniques is used for classifying observations into predefined categories based on multiple variables? A. Principal component analysis (PCA). B. Cluster analysis. C. Discriminant analysis. D. Factor analysis. Answer. C. Discriminant analysis. What is the primary goal of using principal component analysis (PCA)? A. To identify the underlying factors influencing the data. B. To reduce the number of variables while retaining most of the data's variance. C. To predict the future behavior of variables. D. To classify observations into clusters. Answer, B. To reduce the number of variables while retaining most of the data's variance. Which multivariate technique is most suitable for analyzing the relationship between two sets of variables? A. Canonical correlation analysis, CCA. B. Cluster analysis. C. One-way ANOVA. D. Regression analysis. Answer, A. Canonical correlation analysis, CCA. Conclusion. Multivariate statistics provides a powerful set of techniques for analyzing complex datasets with multiple interrelated variables. These techniques are invaluable in biological, medical, and social sciences where multiple factors influence the outcomes being studied. Mastering multivariate methods allows researchers to extract meaningful insights from large and complex datasets, aiding in the development of more accurate models and predictions. You said. Correlation, graphic and mathematical methods. ChatGPT said. Correlation, graphic and mathematical methods. Introduction. Correlation is a statistical measure that expresses the extent to which two variables are linearly related. It helps in understanding the strength and direction of the relationship between variables. In biological and zoological research, correlation can be used to study how two or more variables, such as the relationship between temperature and metabolic rate in animals, or the relationship between feeding habits and growth rates in plants, are interconnected. There are two primary methods for measuring correlation. Graphic methods, mathematical methods. Each method provides a different way of visualizing or calculating the degree of association between variables, which is crucial for the interpretation of experimental data. Graphic methods of correlation. 1. Scatter plot. The most common graphic method to represent correlation is through a scatter plot. In this method, each data point is represented by coordinate pair x, y, where x is the independent variable and y is the dependent variable. The plot displays the relationship between the two variables visually. Interpretation of scatter plots. Positive correlation. If the points generally rise from left to right, it indicates a positive correlation, meaning that as one variable increases, the other variable also increases. Negative correlation. If the points generally fall from left to right, it indicates a negative correlation, meaning that as one variable increases, the other variable decreases. No correlation. If the points are scattered randomly without any discernible pattern, it indicates no correlation between the variables. 2. Line of best fit, regression line. To summarize the relationship between two variables, a line of best fit or regression line can be drawn on a scatter plot. This line represents the best estimate of the dependent variable y based on the independent variable x. For simple linear correlation, the line of best fit is a straight line that minimizes the sum of the squared differences between the observed values and the predicted values. Interpretation of the regression line. The slope of a line indicates the direction and strength of the correlation. A steep positive slope suggests a strong positive correlation, while a steep negative slope indicates a strong negative correlation. The line of best fit helps in estimating values of the dependent variable for any given value of the independent variable. Advantages of graphic methods. They provide an intuitive, visual way to assess the strength and nature of the relationship between two variables. Scatter plots allow for quick identification of potential outliers or nonlinear relationships. Limitations of graphic methods. Scatter plots can only show the relationship between two variables and do not offer detailed statistical measures. The line of best fit provides a generalized view but does not account for variability in data points. Mathematical methods of correlation. Mathematical methods provide a more precise and quantifiable way to measure correlation. The most widely used mathematical methods include Pearson's correlation coefficient, Spearman's rank correlation, and Kendall-Stau. 1. Pearson's correlation coefficient R. Pearson's correlation coefficient, often represented as R, measures the strength and direction of a linear relationship between two continuous variables. It can range from -1 to +1. R equals +1 indicates a perfect positive linear correlation. R equals -1 indicates a perfect negative linear correlation. R equals 0 indicates no linear correlation. Formula for Pearson's correlation coefficient, R. Equals n array summation xy minus n array summation x n array summation y n array summation x2 minus n array summation x2 n array summation y2 minus n array summation y2 R equals n array summation x2 minus n array summation x2 n array summation y2 minus n array summation y2 n array summation xy minus n array summation x n array summation y, where n is the number of data points x and y are the two variables being correlated. N array summation denotes summation over all data points. Interpretation of Pearson's R and GT 0 positive correlation, R and LT 0 negative correlation, R equals 0 no correlation. 0.9 is less than or equal to R and LT 1 strong correlation. 0.7 is less than or equal to R and LT 0.9 moderate correlation. 0.5 is less than or equal to R and LT 0.7 weak correlation. 2. Spearman's rank correlation ρ or RS. Spearman's rank correlation is a nonparametric measure of the relationship between two variables. Unlike Pearson's R, which requires data to be continuous and normally distributed, Spearman's rank correlation works with ordinal data or nonlinear relationships by ranking the data points and then calculating correlation. Formula for Spearman's rank correlation, ρ equals 1 minus 6 n array summation d2 n 2 minus 1. ρ equals 1 minus n 2 minus 1 6 n array summation d2, where d is the difference between the ranks of corresponding variables n is the number of data points. Interpretation of Spearman's rank correlation, ρ equals +1 perfect positive rank correlation. ρ equals -1 perfect negative rank correlation. ρ equals 0 no rank correlation. Spearman's ρ is often used when the data is ordinal or when the relationship between the variables is not linear. 3. Kendall-Stau tau. Kendall-Stau is another nonparametric test for measuring the ordinal association between two variables. It is considered to be more robust than Spearman's ρ, especially when there are tied ranks. Formula for Kendall-Stau tau equals c minus d c plus d plus tx c plus d plus ty tau equals c plus d plus tx c plus d plus ty c minus d, where c is the number of concordant pairs, d is the number of discordant pairs, t tx, and ty represent the number of tied ranks for the variables x and y, respectively. Interpretation of Kendall-Stau, tau equals +1, perfect positive association. Tau equals -1, perfect negative association. Tau equals 0, no association. The Kendall-Stowe coefficient is used when the data involves rankings and may have tied values. Advantages of mathematical methods. Quantitative measure provides a precise, numerical measure of correlation that allows for statistical testing. Pearson's R is very effective for data with a linear relationship and is widely used in biology and other fields. Spearman's and Kendall-Stowe are more appropriate for ordinal data or when the relationship is nonlinear, or the data has outliers. Limitations. Pearson's R assumes linearity and normal distribution, which may not always hold true in biological data. Spearman's and Kendall-Stowe can be less informative in cases where the data is truly continuous and linear. Solved previous year question. Question. A researcher wants to investigate the relationship between the amount of food intake and the body weight of animals. The data is collected from 10 animals, and the Pearson's correlation coefficient is calculated to be 0.85. What can be concluded from this result? A. There is a weak negative correlation between food intake and body weight. B. There is a strong positive correlation between food intake and body weight. C. There is no correlation between food intake and body weight. D. The relationship between food intake and body weight cannot be determined. Answer. B. There is a strong positive correlation between food intake and body weight. Explanation. A Pearson's correlation coefficient of 0.85 indicates a strong positive correlation between food intake and body weight, meaning that as food intake increases, body weight also tends to increase. Multiple choice questions MCQs. Which of the following methods is used to measure the relationship between two variables when the data is ordinal? A. Pearson's correlation B. Spearman's rank correlation C. Kendall-Stowe D. Both B and C. Answer. D. Both B and C. What does a Pearson's correlation coefficient of -0.75 indicate? A. Strong positive linear correlation B. Strong negative linear correlation C. Weak positive correlation D. Number correlation. Answer. B. Strong negative linear correlation. Which correlation method is most appropriate when the data is continuous and normally distributed? A. Kendall-Stowe B. Spearman's rank correlation C. Pearson's correlation D. Chi-square test. Answer. C. Pearson's correlation. Conclusion. Correlation analysis, whether graphical or mathematical, is a powerful tool in understanding the relationships between variables in biological and other scientific research. Graphical methods like scatter plots provide a quick visual overview, while mathematical methods like Pearson's, Spearman's, and Kendall's correlation offer precise measures to quantify the degree of relationship. Mastery of these methods enables researchers to interpret complex data and draw meaningful conclusions in their studies. You said. Carl Pearson's coefficient of correlation, ChatGPT said. Carl Pearson's coefficient of correlation introduction. The Carl Pearson's coefficient of correlation, often denoted by R, is one of the most widely used statistical tools for measuring the strength and direction of a linear relationship between two continuous variables. It quantifies the degree to which a change in one variable is associated with a change in another. Named after the statistician Carl Pearson, this method is used extensively in biological, social, and physical sciences to assess a linear correlation between two variables. Pearson's correlation coefficient ranges from -1 to +1. +1 indicates a perfect positive linear relationship. -1 indicates a perfect negative linear relationship. 0 indicates no linear relationship between the variables. The closer the value of R is to +1 or -1, the stronger the linear relationship between the variables. Formula for Pearson's correlation coefficient. The formula for Pearson's correlation coefficient R is as follows. R equals n array summation xy minus n array summation x n array summation y n array summation x2 minus n array summation x2 n array summation y2 minus n array summation y2 R equals n array summation x2 minus n array summation x2 n array summation y2 minus n array summation y2 n array summation xy minus n array summation x n array summation y, where n equals number of paired data points x equals the values of the independent variable y equals the values of the dependent variable n array summation xy n array summation xy equals sum of the product of corresponding x and y values n array summation x n array summation x and n array summation y n array summation y equals sum of the x values and y values respectively n array summation x2 n array summation x2 and n array summation y2 n array summation y2 equals sum of the squared x values and y values respectively. Steps to calculate Carl Pearson's coefficient of correlation to compute R. Follow these steps. Collect the data. Obtain the paired data points for the two variables x and y. Calculate the sums. Sum of the x values n array summation x n array summation x. Sum of the y values n array summation y n array summation y. Sum of the products of x and y n array summation xy n array summation xy. Sum of the squares of x values n array summation x2 n array summation x2. Sum of the squares of y values n array summation y2 n array summation y2. Substitute the values into the formula and solve for R. Interpret the value of R. +1, perfect positive correlation. -1, perfect negative correlation. 0, no linear correlation. Values between +1 and -1 indicate varying degrees of positive or negative correlation, with the strength increasing as the magnitude of R approaches 1. Interpretation of Carl Pearson's coefficient of correlation. Positive correlation R and GT, 0. If R is positive, it indicates a positive correlation, meaning that as one variable increases, the other variable tends to increase as well. For example, as the temperature increases, the rate of metabolic activity in certain animals may also increase. Negative correlation R and LT, 0. If R is negative, it indicates a negative correlation, meaning that as one variable increases, the other variable tends to decrease. For example, an increase in altitude may lead to a decrease in oxygen saturation in the blood. No correlation R equals O. If R is 0, it suggests no correlation, indicating that changes in one variable do not predict changes in the other variable. This may be observed if there is no relationship between two variables. Strength of the correlation. 0.0 to 0.3, weak positive correlation. 0.3 to 0.7, moderate positive correlation. 0.7 to 1.0, strong positive correlation. Minus 0.3 to 0.0, weak negative correlation. Minus 0.7 to minus 0.3, moderate negative correlation. Minus 1.0 to minus 0.7.